Welcome. Today we are going to learn about the isolation of the bacterial genomic DNA. When we are isolating the bacterial genomic DNA, we have to go through the steps. The steps are the first we have to grow the bacterial culture. After growing the bacterial culture, then we have to pellet out the cells, and that is we have to harvest the cells because the DNA, genomic DNA, is present inside the cell. So we have to separate out the cells from the medium, and then we have to prepare the cell extract. That is, we have to break down the cell wall and we have to release the genomic DNA into the surroundings, and then we have to purify the DNA. After purification, we have to concentrate the DNA. So there are the 4 steps. Okay, so there are the 4 steps for the isolation of the genomic DNA from the bacterial culture. First, we have to grow and harvest the bacterial cells. Then we have to prepare the cell extract, and after preparation of the cell extract, then we have to purify the DNA, and then that DNA we have to concentrate. So going step by step, first growing and harvesting the bacterial culture. Okay, so here, okay, first we have to grow the bacterial culture into the medium, the liquid medium, like the defined medium, like the M9 medium, or the undefined medium, like the Luria-Bertini broth. Okay, after the growing of the culture at the optical density about 1, then we have to harvest the cells, because our genomic DNA, it is present into the bacterial cells. So we have to separate out the cells from this broth, and for that purpose there is a use of the centrifugation. Okay, so here after centrifugation we are getting the pellet of the bacterial cells. Okay, so we have to collect this pellet and we have to discard this supernatant. And now when we have got this pellet of the bacterial cells, then we have to prepare the cell extract. And for the preparation of the cell extract, okay, we have to lyse the cells, and for the lysis of the cells there will be the use of the enzymes. And here the enzyme, most frequently used enzyme, it is the lysozyme. You know that the lysozyme, it is the enzyme which is going to act on what? It is going to act on the cell wall. Okay, bacterial cell wall, it is made up of the peptidoglycan, and the beta-1,4 linkage of the peptidoglycan, it is going to be broken down by the enzyme, and that enzyme, it is the lysozyme. So here there is a use of the enzyme lysozyme. Okay, and then with the lysozyme there is a use of the EDTA and the SDS. EDTA, it is a chelating agent. It is going to chelate out the ions. Okay, so it is allowing the disruption of the cell wall, as well as it is allowing the inactivation of the enzymes which are present into the bacterial cells. And SDS, it is the detergent. Okay, and as we know, detergents allow the dissolution of the lipids. And now here, okay, when you look at the structure of the bacterial cell, okay, so here the outermost covering, it is of the cell wall, then there is a membrane. This dark yellow, it is a membrane, and outer one, it is a light yellow color, it is whom? It is the cell wall. So when we have to isolate the genomic DNA which is present inside the bacterial cell, we have to break down the cell wall and we have to break down this membrane. So for that, okay, for that purpose, okay, there is the use of what? There is a use of the enzyme lysozyme, which is allowing the disruption of the cell wall, and the SDS, which is allowing the dissolution of the membrane, which is going to unstabilize the membrane. And here in this way, okay, in this way, the lysozyme, EDTA and SDS together, they are allowing the preparation of the cell extract. It is allowing the disruption of the cell wall, breaking down of the cell wall, and breaking down of the membrane. So as the membrane, as the cell wall, it has been broken down, all the content which are present into the bacterial cell, it is also going to come out, and all this, it is going to be called as the cell extract. Okay, now after that, what we have to do? We have to remove the cell debris. We don't want the cell debris. Okay, what is required? The DNA, it is required. And to remove the cell debris, again there is a use of the centrifugation. Okay, when we are going to do the centrifugation, here after centrifugation we are getting the pellet. It is of the cell debris, and in the supernatant there will be, okay, there will be the DNA, RNA and the proteins. So here the genomic DNA, it is contaminated with the RNA and the proteins. So the next step, it is what? Okay, what may be the next step? Next step, it is a purification. Okay, it is the purification of the DNA from the cell extract. Okay, so for that purpose, for the purification, the method which is used, it is the removing the contaminants. Which are the contaminants? Contaminants, it is the proteins and the RNA. Okay, removing the contaminants by the organic extraction and the enzyme digestion. And the second one, it is the ion exchange chromatography. So going towards the first method, which is what? Removing contaminants by the organic extraction and the enzyme digestion. So here it is our cell extract. Okay, and now this cell extract, it is going to be treated with the phenol. Okay, that is the phenol, it is mixed with this cell extract. With this phenol, you may use the phenol chloroform. Okay, phenol chloroform, it is nothing but it is the full phenol is to chloroform is to isoamyl alcohol. Now here the phenol which is used, it has a pH, and that pH it is of about the 8. Okay, at the alkaline pH, okay, alkaline pH of the phenol, it is allowing the DNA to be present into the aqueous phase. Okay, if the pH of the phenol, it is the acidic one, the DNA, it may get dissolved into the phenol. So here it is very important to maintain the pH of the phenol, and that phenol, it is equilibrated with the buffer, and that buffer, it is a Trissial buffer. So here there is a use of the phenol, which is a equilibrated phenol, or there is a use of the phenol chloroform. In this also the phenol, it is equilibrated with the buffer, that is, it is a Trissial buffer. Okay, so here there is a mixing. When there is a mixing, okay, when there is a mixing of the phenol or the phenol chloroform with the cell extract, what is going to happen here? There is the denaturation of the proteins. Okay, and when, okay, and then when there will be the centrifugation, after centrifugation you are getting, okay, after centrifugation you are getting the 3 layers. One, it is the organic phase, which is of the phenol. Okay, then there is the interface. Okay, and at this interface there will be the coagulated proteins. Okay, and here it is the aqueous phase, okay, or the aqueous layer, and in the aqueous layer there is a nucleic acid, there is a DNA, and the RNA, it is present into the aqueous phase. Now tell me what you will collect. If you want to isolate the genomic DNA, then which phase you will collect? Yes, you are going to collect the aqueous layer or the aqueous phase. It is going to be collected, and then it is going for the concentration of the DNA. The second method, okay, second method, it is ion exchange chromatography to purify the DNA from the cell extract. Ion exchange column, and here there is a presence, okay, this column, it is filled with the ion exchange resins, which are, okay, which are positively charged. Why it is a positively charged? Because your DNA, okay, because your DNA, it is a negatively charged. Okay, so here, okay, these are, okay, these blue colored, it is the resins. Okay, so when you are going to pass your cell extract, okay, when you are going to pass your cell extract to the column, at that time this positively charged resins, it is allowing the binding of the DNA, which are the negatively charged. As it is an ion exchange chromatography, it is totally based on the charges over the molecules. So resins positively charged, DNA negatively charged. Okay, so the DNA, it is getting bound to the resins. Okay, here you are allowing the interaction between the DNA and the resins. Okay, so here, okay, so when there is a completion of the period of the binding, then you have to go for the elution. And when you are allowing the elution, at that time you are going to increase the concentration of the salts. Okay, so during the elution, when you are allowing the increase in the salt concentration, at that time in the eluent, okay, in the eluent you are getting the proteins and the RNA. So here in this column, okay, in this column you are only allowing the genomic DNA to be present into the column, okay. And when you want to elute out the DNA also, at that time you are again going to increase the salt concentration, and when you are increasing the salt concentration, this interaction it is going to be weakened, and the genomic DNA it will get into the eluent. Okay, so there will be the elution of the DNA when the salt concentration it is going to be a higher one, and here you are getting the DNA into the tube into the eluent. Okay, then this eluent it will go for the concentration of the DNA. So how to concentrate the DNA? This concentration of the DNA it is done with the 100% ethanol. Okay, so here there will be, okay, in the tube, okay, like here. Okay, here it is what it is your solution of your DNA, and you are going to layer the 100% ethanol over it. So what will happen? Okay, what will happen here? The buffy layer it is going to be formed, and that buffy layer it will be of the DNA. Okay, at this interface there will be the concentration of the DNA, and if the concentration of the DNA it is a higher one, just by putting a rod, like here it has shown this, it is a rod, it is a glass rod, and that glass rod it has been dipped into this solution, and then by just swirling, okay, there will be the there will be the taking out of the DNA from this solution. The second method, when the concentration of the DNA it is a lower one, okay, at that time what is going to be done? You are going to put the 100% ethanol, and you are going to mix it, okay, and you are going to keep it at 0 degree or -20 degrees Celsius, and allow the DNA to get precipitated. Next day, what you are going to do? You are going to centrifuge it, okay, or after 3 to 4 hours or 1 to 2 hours, you are going to centrifuge it, and after centrifugation, here you are getting a pellet, okay, and that pellet it is of the nucleic acid. Okay, here there may be the contamination of the RNA with this pellet, and that contaminated RNA it is going to be removed with the help of the treatment with the RNAs. So the RNAs it is going to cleave the RNA, and now this pellet it will be with whom? It will be with only the pure genomic DNA. Okay, and here to remove the other contaminants, you can do the washing of this pellet with the 70% ethanol, and then you are going to dry it, and then you are going to dissolve into the buffer, and that buffer it is a TE buffer. Okay, so in this way there is the isolation of the genomic DNA. If you have liked my video, please subscribe my channel, and also share my video with your friends, so the knowledge it is going to spread. Thank you.